and with the title Where Next, which means that I will just give briefly an introduction into the field of super-resolution microscopy, with special focus on single molecule localization microscopy, a few examples really just briefly, and then we'll introduce to you a few fields we started to investigate 2-3 years ago, which is receptor imaging and quantification, and the use of this technique in personalized immunotherapies, as well as genetic code expansion, in other words, to use unnatural amino acids for labeling of proteins of interest. So, about 20 years ago, several groups started to find ways to bypass the diffraction barrier of light microscopy. And these efforts have been rewarded 2014 by the Nobel Prize in Chemistry given to W. E. Murner, Eric Betzig, and Stefan Held, and finally. So the trick behind all these different techniques which have been introduced is that you either confine the fluorescence emission of fluorophores to a specific area only, or that you temporally, in time, separate the fluorescence emission of single fluorophores stochastically. And this is the trick behind single molecule localization microscopy, so that you temporally control the fluorescence emission of single fluorophores. So just to introduce this technique, because there will be another talk given also using also a single molecule localization microscopy this afternoon given by Mike Heilemann. So to introduce this technique, just a single slide. So imagine you have 1 square micrometer and you attach a fluorophore every 50 nanometers, so you end up with 2,500 fluorophores in this square micrometer. And if each of these fluorophores emits fluorescence, you will detect each fluorescence signal of these emitters as a blurred point spread function with a diameter of about half the wavelength of light. And if they all emit simultaneously, you would end up with such an image. However, now imagine that it would be possible to separate the fluorescence emission of these individual fluorophores in time in a way that ensures that out of these 2,500 fluorophores, only a few of them are allowed to show or to emit fluorescence, let's say 2 or 3, at any time of the experiments, while 2,497 or 98 fluorophores stay in an off state. So that in each of these frames, while you record this video, there is only a sparse subset of 2 or 3 fluorophores in active state. And then you can localize the positions of the center of mass of these signals by approximating the signal using a 2-dimensional Gaussian function. And finally, if all of these fluorophores contributed, so which means to the signal that they stayed at least once while you recorded your video in a fluorescent state, you can finally reconstruct an image out of that. So the trick to do that was that time then 2005 only to find ways to switch or photoactivate fluorophores, and this is where these different acronyms come all into play, like PALM and FPALM and STORM and DSTORM, and the difference between STORM and DSTORM then was just simply that the group of Xiaowei, Zhuang in Harvard and my group at that time together with Mike Heilemann in Bielefeld, that we published simultaneously in 2005 a paper that you can switch organic fluorophores by means of light in the presence of some special buffers. It was a thiol buffer, and both papers appeared simultaneously in the same week even, had been published. The only difference between the two papers was that in the PRL paper it was claimed that you need an activator and a reporter dye, so a pair of fluorophores. We published that it works for any dye, so you can simply buy your antibody labeled with a fluorophore, add this buffer, and do super-resolution microscopy. Then the following years we investigated in detail the switching mechanisms. I will not go into detail today because then I would run out of time. The only thing I can say is that this thiol buffer you're using transfers the fluorophores into a reduced state, a radical anion state, or sometimes also a fully reduced state with a surprising stability. And then the fluorophore is pushed back to the fluorescent state by oxidation with molecular oxygen naturally present in your solution. And by having these tools now in hand, or these tricks, you can now simply buy a labeled phalloidine. It's a toxin binding to aptine, fix a cell, add the phalloidine, wash off, and then add this switching buffer, 100 millimolar of thiol, mark up to ethylamine, and then these are raw data. You record this video and you see that only a sparse subset of all these fluorophores is in an active fluorescent state at any time of the experiment, and that the distance between these point spread functions is large enough, which allows you to fit these point spread functions using or approximate the center of mass using a 2-dimensional Gaussian function. And then you end up, after some time, 11 million localizations in a super resolved image. And this can be done, as we just recently showed together with people from Genelia Farm, with Luke Levy's group. This can be done not only with organic dyes and photo-switching cover, but for Luke Levy's designed and developed also some photoactivatable dyes, so they are non-fluorescent unless you irradiate with low intensity of UV light, and then these bonds cleave off, and the fluorophore reforms, and then you have to photo-bleach the fluorophore, localize it, photo-bleach, activate, localize, photo-bleach. So it takes some time, this image, about 6 hours here. It's a very slow technique, but then you end up with a localization precision of 5 nanometers. A few other examples, as I told you, of course, then you can buy different antibodies directed, for example, against different enzymes of the respiratory chain on the inner mitochondrial membrane, ATPases and so on, and then visualize the distribution of these enzymes in the mitochondria. And a few examples what we have done in the last years are shown here, together with Martin Eilers. He's an oncologist in Würzburg, so he is interested in the changes in mitochondrial structure upon tumor development. So we published a paper by using D-storm super-resolution microscopy in cancer cell, and Thomas Rudel, who is a microbiologist, where we investigated the invasion or the infection of these cells by chlamydia in rat, and how they then fuse together, these bacteria, surround the mitochondria and destroy the substructure of mitochondria. It looks quite different than these images. The mitochondria are destroyed by these bacteria. Okay, what else have we done in the last years? In many other groups, we established then not only cellular imaging, we also established 2D, 3D localization microscopy in tissue, like, for example, here in brain slices, 24 micrometer thick brain slices, and we can reconstruct finally together with the groups of Manfred Heckmann and Annalena Sirene in Würzburg, neurobiologists. We can do it even sequentially in 2 colors, 3D D-storm of some synapses which interact with these boutons here, labeled with GFP, and the nanobody directed against GFP, and in this case, the active zones have been labeled with the active zone marker bassoon and an antibody directed against this bassoon. So we are able now to distinguish different synapses and code them depending on their size. In red are the bigger synapses, in blue are the smaller synapses, and these are the boutons we are interested, for example, in, and then we can see which synapses interact with these boutons, and depending on where you are in the brain, you will see that the active zones are larger or smaller, for example. And how many proteins are there? You get numbers, simply about how many antibodies have bound. Not numbers directly about the proteins, but at least about the numbers of proteins which bind to these proteins in different areas of the brain. Okay, I promised that I will talk more about where next. And where we started a few years ago is quantification of plasma membrane receptors proteins by D-storm. So why receptors? Receptors are key regulators of life. You're all aware of the importance of receptors. About 90% of all drugs available today are directed against receptors, and the entirety of all receptors are called the receptome. There are different functional systems which receptors control, like growth, vascular tone, synaptic transmission, metabolism, host defense, and they all are related to severe diseases like cancer, hypertension, and so on and so on. We all know these drugs. And just recently, and yesterday, the Nobel Prize has even been awarded for people working on cancer immunotherapy. Receptors are, of course, of fundamental importance for immunotherapy. Okay, so receptors are the target for the pharmaceutical industry. They are super important for signaling, cellular communication, and all the things we're interested in. Most of these things, however, despite of these important, also for clinical applications, there's not much known about their expression rates, about the distribution of these receptors on different cells and tissues and their interactions. And this has been then the starting point to address this problem in the future, the receptome of a cell. Can we really investigate and quantitatively determine the receptome of cells, of different cells? Just historically, a few words about this, which was very confusing for me as a chemist and physicist. This CD nomenclature which has been introduced in 1984 by immunologists who met in Paris, and this is what they do every year now, these human leukocyte differentiation antigen workshops. And different labs had different antibodies binding to different cells, and so they agreed in 1984 that they shared the different antibodies, and if different antibodies bind to the same cells identified by other means, then they call it a cluster. Not really a cluster of receptors, like we talked today, a nanocluster. It's just a cluster of antibodies used in different labs that they all bind to HeLa cells on the plasma membrane. And now we have these 4,000 different surface molecules, and we have CDs ranging from CD1 to CD371. And how would he know all the knowledge which exists today? Where does it come from? Mainly from flow cytometry and some immunocytochemistry imaging. And in most places, at least in hospitals, and we tested this in Berlin, in Düsseldorf, and in Würzburg, in the university hospitals, these flow cytometers have a sensitivity of about 1,000 receptors per cell. Not cells. 1,000 receptors, 1,000 receptors per cell, but of course they can measure about 1,000 cells per second. So the sensitivity is severely limited, and all the knowledge we have about expression rates of receptors, whether they're here or there, and we determine the developmental stage of a cell depending on these expression profiles, are, not useless, but limited. So if we now want to really determine the receptome of, let's say, a uricot cultured cell, let's start like this, and these are the people who did that in the work, in the group. Sorry. This happens if you do not provide a photo, if you enter my group. So we do not now really know which receptors are expressed on cells, even at low expression rates. So we step back, or we looked into literature. We have been looking for mass spectrometry data, where people published lists of proteins or receptors found in the plasma membrane of different cells. And this is one of these papers. And so we started with these receptors which should be expressed in uricot T cells, and with these two which should not be expressed. Or it's controversially discussed, low amounts you find always. We established a protocol, and what you do not want to have is that if you label live cells with antibodies, then these antibodies will induce internalization of the receptors. If you fix the cells before labeling with antibodies, antibodies will penetrate the cells and label also intracellular receptors, but if you are interested in the membrane receptome, as we heard in the morning, so you have to avoid these processes, so we label at 4 degrees centigrade to avoid internalization. And then we add the antibodies, and after washing, we fix and do this super resolution microscopy imaging, and then it looks typically like this. So for some of these receptors, you see clustering in 2-color experiments, single-color experiments. And now you might wonder, CD3 shows clusters, and just recently there was a paper in Nature Immunology by Gerhard Schütz's group who studied especially these clustering artifacts in this kind of measurements on primary T cells. But still we see CD3 clusters on these uricot T cells, but this is simply explained because uricot T cells, cultured T cells, they express interleukin-2, and this activates these cells. If we do the same experiments on primary T cells, using the same antibody, there are in fact no clusters. Yeah. Just to convince you. What else do you have to do? You have to ensure that you saturate all the accessible epitopes with the antibodies, so you have to titrate antibody concentrations. And then you have to compare different dyes. We are doing 2-color experiments in most cases with Alexa 532 and Alexa 647, the red and the green dye. But if we, for example, use the same antibody, addressing binding to the same receptor, we do the same experiments, under similar experimental conditions, we find that using just another dye, we detect half as many localizations for Alexa 532. So the only reliable data, we know this also from from correlative EM and D-storm data at the nuclear pore complex, that this dye provides reliable information concerning localization numbers, and most other dyes, unfortunately not. So we have to do all the experiments with Alexa 647. Next problem, how many receptors you see might depend on which antibody you're using. Maybe some epitopes are better accessible for this antibody and others for that antibody. So we did also these experiments using different monoclonal, naval polyclonal antibodies, but for most of these receptors, for example CD5, we always find the same, which is good. Yeah, really good. and also depending on the cell cycle state. So we have to do averaging and a lot of work to come up with some reliable numbers, and these are the numbers for the joker T cell. So the lowest expression is here for CD4, which is about 1 receptor per square micrometer, and for the negative controls we find something below 0.01 receptor per square micrometer in these experiments. If you then are still interested in doing multicolor experiments, you are of course restricted by this problem that different dyes deliver different numbers. So, but there is a way out of this problem, which has been introduced by Ralph Jungmann's group in Germany at the Max Planck Institute in Munich, which is called DNA exchange storm. So instead of attaching a dye to the antibody, you attach a short oligonucleotide to the antibody. So you use 3 antibodies, attach 3 different oligonucleotides. You label at 4 degrees centigrade simultaneously. You wash, you fix, and then you add a complementary oligonucleotide for antibody 1, and do this D-storm imaging. You wash off with formamide, add oligonucleotide 2, and by that you can address the different antibodies via the DNA sequences, and it has never been shown whether this can be done quantitatively. And if we compare the data from single color measurements to 2 to 3 color measurements using DNA exchange, the numbers are at least comparable. Okay, so there are other problems, of course. So one problem is, of course, cells usually don't live isolated. They interact with tissue. And the other problem is most of plasma membrane studies, at least by light microscopy, have been performed using total internal refraction fluorescence microscopy. Well, simply due to the fact that the signal-to-noise ratio is the best. Yeah. Of course, you can use this high-low, this highly inclined optical laminated sheet, so a dirty turf, and also irradiate the apical membrane. But the easiest and the most beautiful thing you can do is if you could do whole cell imaging of receptor distributions, and this is what we established then. We built a lattice light sheet microscope in the group. Felix did that, and these are raw data. So if a cell moves through the lattice light sheet, and you detect all this localization data by 3D astigmatic D-storm imaging, and if you then do this imaging, you can come up with the distribution and can visualize the distribution of these receptors in 3D. And you see some antibodies non-specifically bound. This is the cover slip site then, on the cover slip, but the distribution doesn't change. So there is no clustering artifact due to contact with the cover slip, for example. And we have also other examples. This is for CD2, also expressed at high rates, and you can investigate the cell-cell contacts, whether there the distribution of receptor changes, for example. And this is for CD3. Astrophysicists might see something different, but this is for CD3 clustering, and you see that the clusters are not influenced, apical, basal, or cell-cell contact. Okay, so what's it good for? And this is, of course, good for what we had in mind, which is cancer immunotherapy. And there we started a collaboration 2.5 years ago with the director of the University Hospital in Würzburg, Hermann Einzel, and his group, Michael Hudicek and Thomas Nerota, in investigating the receptor distribution and the expression of tumor-associated antigens receptors. So there are different ways of immunotherapy, and one of the immunotherapies which have been invented in Würzburg is this bispecific antibody therapy. The drug on the market is blinatumomab. So it's a CD19 antibody fused to a CD3 antibody. And if the CD19 binds to leukemia cell, lymphoblastic leukemia, and on the other side it can then bind to T cell, activating the T cell, and this way the T cell is located, brought in close contact to the CD19 positive cell, and then it will kill the cell. Another immunotherapy which is getting increasingly important is CAR T cell therapy. And here we have one of the biggest centers of Europe here in Würzburg. So what are CAR T cells? These are modified T cells. We'll come to it in a minute. But this drug has also some side effects people discovered. I will also come to that in a minute. So what is CAR T cell therapy? You take the T cells out of the patient, You take them in culture, and then you transfect the cells that they express, I will say it in simple words, fat fragments directed against CD19 for acute lymphoblastic leukemia. So you have now these antibody recognition sites for CD19 on the surface of the T cells, and then the T cells are reinjected into the patient, and then these T cells bind to all CD19 positive cells and kill these cells. Yeah? And this was of interest to us, especially due to this publication in New England Journal of Medicine, the Holy Grail Journal of Medical Doctors. So I wasn't aware of it before, with an impact factor of about 70, and a paper from a group in the UK who tested CAR T cells directed against these lymphoblastic leukemia cells against CD19. They treated multiple myeloma patients, just for fun, because it is known that multiple myeloma cells of patients do not express CD19, according to the knowledge today, but they succeeded in 50% of the cases. And this was then the starting point that we said there is something wrong. Yeah. And the other point is, of course, these reports, you always find that people pass away under this therapy because there are some severe side effects. So what have we done? So we started with 20 patients in the hospital, multiple myeloma patients. And then we started first of all, of course, we did a quantification with a cell culture line expressing CD19, compared flow cytometry experiments and quantification with our data and everything. But then we started with 20 patients, and these are some flow cytometry negative cells, multiple myeloma cells of this patient. You first tested for CD38, just white field versus microscopy, and CD138, and you see both cells here are positive, so this is a tumor cell most likely. And then you test for CD19 by high resolution here, D-storm, to quantify the numbers of signals you see. And this is a typical signal. This cell would be negative in all flow cytometers in any hospital. Yeah? And this is an example of a negative cell. Yeah. Okay. Then you do quantification for different patients. And you end up with a bimodal distribution of receptor densities on these cells. On these multiple myeloma cells, something around 1 and something around 0.023, something like that. Okay. Then you test other antibodies, because you can never be sure. Maybe these antibodies bind non-specifically on these cells. Maybe there is no CD19 expressed. Yeah. So another isotype antibody doesn't bind officially, so this population disappears, this lower receptor density population remains. And then we did the following. In order to really ensure that these are really CD19 receptors we detect on the cell, we treated these patient cells ex vivo with the T cells of the patient. Nothing happens, we still have two populations for all the different patients. But then we treated these cells ex vivo with modified T cells of the patient. So we transfected the T cells that they express exactly the CD19 binding fat fragments on the surface that they should now bind to the cells and kill them. And if we do the experiment afterwards, the second population disappears, which tells us that this is not non-specific binding, that there is all the CAR T cells bind as well non-specifically. Yeah? A lot of statistics with flow cytometry data and D-storm data, and for example for a high expressor here, which is really also in flow cytometry, 20% of the cells are positive, so from time to time you find these patients, but most of them are, even though they are positive, they are negative in flow cytometry, and for other cases, like here, this patient where this population disappears after CAR T cell treatment completely as well, for this patient, which convinces us that this was really CD19 expression on this multiple myeloma cell. So to summarize, in this first study we finished now a year ago about, we investigated these patients. Finally, for those we could collect enough statistics to publish the data. And we saw 3 of them would have been tested positive by flow cytometry, so they can potentially receive this immunotherapy. All the others would have never received the immunotherapy because they are FAX negative in our hospitals. but they express these receptors. Yeah. So this is the summary at this point, so that I'm convinced that super-resolution microscopy and the single molecule techniques, in some cases for really low expressing receptors, for low expression rates, you even don't need super-resolution microscopy. You simply have to do single molecule sensitive wide field imaging. Only for these higher expression rates, super-resolution microscopy, single molecule super-resolution microscopy helps. We could also, another very interesting thing we did studies, so how many receptors are required in order that such a CAR T cell kills the cell? It's about only 10 receptors for these CD19 CAR T cells. So this can potentially explain many side effects. Yeah. So why not simply lowering the aggressiveness of these cells so that maybe 100 cell receptors are required? Yeah? This could make the therapy much safer, for example. This is what people are working now together with us. This I already said, so I think that these new super-resolution techniques can be used advantageously to improve the efficiency and the safety of personalized cancer immunotherapies. And this is what we're doing in the moment. We even started to work with tissue imaging with high sensitivity. So this is something coming up as well. It's that you visualize the distribution of some tumor-associated receptors or antigens in the plasma membrane by using an antibody, or in most cases a peptide in this case. And then you add isotopes like gallium-68, which emits positrons. And then you inject these antibodies and they bind to these receptors, and then you detect where are these antibodies sitting. This is for prostata-associated membrane antigen, and you see all these metastases in the body. And then you take the same antibody, but this time you attach another isotope, lutetium-177. This is a beta emitter, which kills locally, of course, beta emission. These cells, you inject it, and by this you hope to destroy this metastasis. But nobody in the hospital who is using all these techniques has ever seen whether the tissue, the tumor material, or the cells are really expressing these receptors. And this is what we do for them in the moment, and hopefully you find something interesting. So like here for CXCR4 on a primary adrenal tumor tissue of patients. Meanwhile, we also have primary brain glioblastoma tissues, not paraffin embedded, which is difficult to get. And another example is shown here that this is another therapy. This is a commercially available anti-multiple myeloma antibody directed against CD38. It's just an antibody. It's called daratumumab. And these are some patients before the treatment with this daratumumab in the hospitals. And this is the receptor profile or the expression rate after treatment, or when the treatment starts, because people discover in the hospitals that after a few days or weeks of treatment with this immunotherapy, they are no longer sensitive. So the receptor expression rates decrease. But this is not the case. I do not have the results with me, but our preliminary data simply show that this antibody binds so specifically and so stable to these receptors that even after days, they are still on these cells. If you use other antibodies directed against a different epitope, you still see higher expression rates even in these patients. Okay, the last 2-3 minutes, I'm now at 30 minutes. I will share with you some results of what we did concerning changing the methods how we label our proteins of interest we want to visualize by super-resolution microscopy. Of course, in these primary materials, what else can we do than using antibodies? There is not much alternative. There are not many alternatives. But for other proteins you're interested in, it might be useful to use a smaller probe. Antibodies, IgG antibodies, have a size of 10 nanometers. If you're using primary and secondary antibodies, you end up with 15 to 20 nanometers. Now, assuming the best spatial resolution we can achieve of about 20 nanometers, this is kind of contradictory. So people in the lab, Gerti, Alexander, and Franziska, they started to use this unnatural amino acid labeling. So it's just that some bacteria found a way to incorporate an amino acid at the stop codon, the UHG stop codon, and you can now incorporate this modified amino acid exactly at this position using a special tRNA synthase. And if you do that, for example, if you label NMDA receptor, in this case in hex cells, we always start with hex cells, and you attach a GFP so that you always see if there is GFP expression, then your unnatural amino acid has been incorporated. Then you can visualize the GFP intracellular and also at the membrane. You can visualize the NMDA receptors by a monoclonal antibody in the plasma membrane emerge, but you can also visualize the receptors by click chemistry, just clicking a diagram to the extracellular receptor domains with a higher efficiency than using antibodies. And then you can do super resolution microscopy and come up with images, which are, of course, depending on the expression rate, you transfect the cells, transfection efficiency, and all the other things. The next thing you have to demonstrate is that these receptors are still functionalized. So this has been done by Ulrich Terputz, who is doing electrophysiology, to demonstrate that these NMDA receptors are still okay. They do their job. And I will stop with this transparency just to show that it's much more efficient, the labeling with click chemistry, than the antibodies, because of steric hindrance and maybe other problems associated with the antibodies. And these are the projects we are currently working on. These are the receptors we are labeling together with in different collaborations. Kinase receptors, GABA, AMPA receptors, adrenergic receptors, GPCRs, and so on. They all work. We have established this now and can do nice studies about single molecule tracking in the membrane and other studies together with these people. Finally, I have to thank all the people. I hope that I didn't forget to mention most of them on the slides who do the work in my lab, and I also have to thank the people. I thank the people who gave us the money, and I thank you for your attention.
Thank you very much. Talk is